So the next we are having that is the classification of the enzyme into the substrate. Okay, that is based classification. This is based on substrate. Okay, so after that, as you guys all know that we are done with the reaction type, so we are having four groups of the substrate. Number four groups. Number one we are having that is proteases. Proteases, what does it mean? It means that these are basically the enzyme that basically act on proteins. For example, pepsin, trypsin. These basically digest the large, you can say that polypeptide means large macromolecules into the monomers or small polypeptide. Okay, these basically the enzyme. This pepsin basically work in the stomach, and this trypsin basically work in the small intestine. So these both enzyme basically having the same function, that basically act on protein, and they basically the large protein basically convert into the small proteins. Okay, after that we are having the more enzyme. We are having amino peptidases, and we are having that is carboxypeptidases. Okay, these are basically the enzyme. They basically these small polypeptides, they basically convert the small polypeptides into the peptones or dipeptide. Okay, after that we are having the next enzyme, that is erepsin. That basically the enzyme, basically the dipeptide, they convert the dipeptide into the amino acid. So finally the product we are having, that is amino acid, the monomer amino acid. So the large, you can say that the protein basically convert into the amino acid with this help of the enzyme. We are calling that as a proteases enzyme. So this amino acid basically is dissolved into the cellular activities. So this is the amino acid. After that we are having that is lipases. What is lipases? Lipases like the macromolecule like lipid. We were having the protein. This basically is the lipid that basically change in the fatty acid. Likewise, as you guys all know, the protein was changed into the amino acid, like polymer was changed in the monomer. So this polymer is changed into the monomer. So this the enzyme which act on the between this is the lipase, pancreatic lipase. This is the enzyme that basically convert the lipid large molecule into the small molecule. So this will also dissolve in the cellular activities. After that we are having the enzyme that is carbohydrases, which means that we are having the enzyme that work on like you can say carbohydrate. So likewise we are having the amylase or dietase. This basically digests the starch that we are eating through food, that basically convert the starch or glycogen into the maltose. Amylase is basically in the enzyme which is in our mouth. So the starch we are eating, that will convert into the maltose. After that we are having the enzyme that is cellulase. The cellulase basically convert into the cellulose. This is basically a disaccharide. After that we are having the maltase. As you guys all know, this basically maltase digests the maltase into the glucose. So after that we are having the sucrase. This is basically sucrase is basically the enzyme that basically act on sucrose molecule, and sucrose convert into the glucose and fructose. Okay, after that we are having the enzyme that is sucrases. After that we are having lactase basically the enzyme basically that digests the lactose molecule into the two molecule of, not two, but glucose and galactose. Here the maltose is basically converted the 2 glucose. Yes, so this is the enzyme basically convert the product like molecules into the small molecules. Okay, after that we are having the nucleases. What are nucleases? Nucleases are basically involved in the breakdown of the RNA and DNA. For example, RNAs, okay, DNAs, ATPase. Okay, so the as you guys all know, the RNA, this basically RNA's enzyme basically digests the RNA into the ribonucleotides. As you guys all know, nucleotides we are having the you can say that the chain of the carbon. Okay, on the first carbon we are having the nitrogenous base, and last carbon we are having the phosphate group, phosphoric acid, sorry. So this is the nucleotide. So that RNA basically convert into the ribonucleotide. Likewise, the DNA will convert into the DNA deoxyribonucleotide. Okay, after that we are having ATPase. Likewise, so they basically the ATPase basically enzyme is the enzyme basically hydrolyze the ATP in muscles. So this was the substrate, you can classification that is based on substrate. Okay, so these are the group of enzyme basically work on the polymers to the convert into the monomers. So this was